Okay, hi everyone. Grab your tea because we're going to talk about a new publication that just came out. Uh, just after that last publication, we talked about how a different cannabinoid could be effective against SARS-CoV-2 virus. There are some similarities between these publications, but there are a lot of differences. So we're going to dive into exactly what those are. Okay. So as always, I am a recovering academic, so I am going to use my PowerPoint. This is the paper we're talking about. It's in Science Advances, which is a great journal, and it's titled Cannabidiol Inhibits SARS-CoV-2 Replication Through Induction of the Host ER Stress and Innate Immune Responses. If you need help figuring out how you can read this, you can start by just Googling it, but in my last video, I describe it a little further, how you can get the supplementary information, and how you can download it as a PDF. If you also want to follow through some of the figures with me, you could pull this up. I'm going to show one or two of them, but not all of them. Quick background information that's super relevant for this publication that was a little less relevant for the last publication. What is so important that we know is that when we're talking about genes in this publication, just know that that gene codes for a protein. So it's super relevant, not just because the gene is activated or not activated, but because if that gene is not activated, it's not producing that protein. If that gene is activated, it is producing a protein. And the proteins are essentially everything that's causing our reaction in our body. So this is essentially called the central dogma of biology. And that's just a little bit of background information I wanted to give just to make sure we're all on the same page. Okay, so similar to the last paper I was discussing with cannabinoids and COVID, they started with cell data. And just a reminder about cell data, these are isolated cell lines, meaning it's a single type of cell that's grown in an incredibly controlled environment in a laboratory so we can study individual variables. We are unable to study every single cell in our body at the same time because the amount of variables we'd be looking at would be astronomical and we would never be able to actually come up with any conclusions. So these data were taken on isolated human carcinoma lung cells that express the ACE2 receptor, which is the receptor that the spike protein binds to in order to infect cells. So just a reminder, your cells in your body are expressing that ACE2 receptor, and the spike protein is expressing, well, the COVID virus is expressing the spike protein, which has different segments. That spike protein will bind to the ACE receptor, and that will facilitate the actual infection. The reason we're using essentially cancer cells is they're immortal. They can live forever. And that is the problem with cancer, but that's also how we figured out how to grow cells in the laboratory. Since they can grow forever, we can keep using them and keep testing them. So that is very common practice. Essentially what they did here is they pre-treated the cells, these human lung cells, with CBD, gave them a little bit of time, and then they exposed the cells to the SARS-CoV-2 virus. This would be kind of equivalent to you taking CBD every day and then getting the COVID virus in your body. Okay, so they're pre-treating it. After 2 days, they were monitoring if the cells were expressing the viral spike protein and just measuring the amount of viral load. Another little bit of background information here before we talk about the results further is when we ingest CBD, it is metabolized in the liver and it produces a molecule called 11-hydroxy-CBD. The only difference is this circle in red here showing this OH group, which is called the hydroxy group, which is why it's called 11-hydroxy-CBD. You might think these are so similar that they probably act similar in our body, but they don't. The same thing happens with THC. When you ingest THC, it is metabolized in the liver and produces 11-hydroxy-THC, which is actually more psychoactive and makes you feel more high, which is why edibles probably make you feel a little bit different. So this is really, really important that they tested this. So they weren't only testing CBD in these cell assays, they were also testing 11-hydroxy CBD. And when I say assay, that means a laboratory test. It's a test, an assay. They're assessing something. Okay, so the relevance here is that when you take in CBD, when it's circulating in your body, some of that is going to stay as just CBD, but some of it is metabolized into 11-hydroxy CBD, which is then the actual molecule that's interacting with your cells, not the CBD. So it's very important they tested this to see if us humans were taking CBD as a preventative against COVID and it's metabolized, is that metabolite also going to protect us from COVID? And the answer is yes. In their assays, both CBD and 11-hydroxy CBD were effective in preventing against SARS-CoV-2. But no other metabolites were active. So in the last publication, we emphasized that CBDA and CBGA were the most active compounds. They never tested CBGA, but they did test CBDA, and they found that was not active. We're going to compare and contrast the two papers at the end, but something that is really important here to know is that there are multiple druggable targets when we think about the COVID-19 virus. Both papers were testing different things, so they're not like competing against each other. One paper's data isn't invalidating the other paper's data. Instead, they're saying, okay, if we wanted to target the spike protein, like the last paper, these compounds could be effective. And this paper, they're saying they didn't find compounds that were effective against the spike protein, but they're finding that some compounds are effective in other ways at preventing the COVID-19 virus from spreading throughout the body. Okay. Now, something, again, that's different than the last paper is the effective concentrations for the results to be meaningful in these studies. The last paper, the concentrations were pretty high. And this is a little bit difficult if we're not used to seeing these concentrations. But the last paper, it was about 21 micromolar. And micromolar is a concentration, okay? So in this test or this assay, for the cell data, they found the effective concentration for 50% of those cells to essentially be effective against COVID with the pretreated CBD to be around 1 micromolar. So this is significantly lower than the last paper. And not only is it significantly lower to inhibit that viral replication, it's also much more reasonable. And they also demonstrated that it's reasonable enough that we have data that it's possible in humans. So I just want to also emphasize they confirmed the results from their first cell assay results with 2 other cell lines, including another lung cell line and a monkey kidney cell line. So they demonstrated that it is possible to get the plasma levels of CBD and 11-hydroxy CBD to just about the doses that they were finding to be effective. How did they do this? They did this because a lot of people take CBD regularly. Some people take it at home just for their, just to maintain their body's homeostasis, their endocannabinoid system. But some people need to take CBD for seizure medication to prevent seizures. That medication that is FDA approved and you can get it through your pharmacist is called Epidiolex. Okay. They were taking really high concentrations of Epidiolex, but that's not uncommon because CBD is very safe in the body and super well tolerated. So they were taking 1,500 milligrams per day for 7 days. And after those 7 days, they found that the CBD levels in the plasma were 1.7 micromolar and the 11-hydroxy CBD, which is the metabolite after it's metabolized in the liver, were 0.57 micromolar, which is within the therapeutic range to inhibit SARS-CoV-2 in humans if we were only considering the cell data here. So that's really, really promising. And also keep in mind that all the data we're talking about today is what we call preclinical data. When we look at cell data, when we look at surveys, when we look at animal data, this is all preclinical, meaning all of this data is going to help us actually get into humans, study this in humans, because it costs over $1 billion to do an entire human trial. So all this data is saying, okay, we have enough evidence to now move forward and test this in humans, which isn't scary because we know how many people already take CBD and we know it's really healthy. Well, healthy. We know it's not harmful to you. And this would then set us up for another study that's specifically looking at the outcomes of the SARS-CoV-2 virus and the COVID-19 infection. So I just want to show you the figure of what this looks like. I believe this is figure 1. I mentioned that the EC50 was 1 micromolar. So you can see on this graph, this is about when this chart's, you can kind of see the bend in the chart. That's how they come up with the 1 micromolar. There's actually tools we can use to find the definitive amount here. But again, that's a very reasonable concentration. And here on the right in panel B, you'll see supplier A, B, C, D in reference. This is actually testing the purity of some of the CBD products that they had purchased from different retailers. This tool here that they're using to measure the purity is called NMR, nuclear magnetic resonance. And it's essentially looking at like every proton in that molecule is going to give a certain pattern based on the arrangement. We can see that all of these look incredibly similar. All of those peaks kind of line up with each other, meaning that all of the suppliers that supplied the CBD, it was a very pure compound, which is good. At the end, they're going to emphasize the importance of the purity of the CBD. Okay, so the results from this paper we're talking about did confirm the results of the last paper we were talking about by Bremen and colleagues, saying that CBD does not prevent the viral entry into the human cells. Remember, the last paper was talking about how CBDA and CBGA could, but CBD was ineffective, and these results totally confirm that. But this paper is emphasizing that CBD is effective, 95 to 99% effective, at inhibiting the SARS-CoV-2 spike protein expression in the host cells after it has entered. So this means that CBD acts post-entry. So that COVID virus, the spike protein's still able to bind to the ACE2 receptor. It's still able to enter your cells, but that is when the CBD starts working. Okay, so then we're going to now talk about, I believe this is figure 2, I could be wrong here, but RNA-seq or RNA sequencing of the lung cells pre-treated with CBD then exposed to the SARS-CoV-2 virus. So let's just talk a little more background on that. RNA sequencing, so they're able to, whatever RNA sequence of interest that they're looking for, they're able to actually look at that, look for that, and determine if it's upregulated or downregulated based on these data. And remember that the cells were pre-treated with CBD, then exposed to the SARS-CoV-2 virus. So they found very significant suppression of the SARS-CoV-2 virus, induced changes in gene expression. What the heck does that mean? It means that when that COVID virus enters your cell, it starts to change some of the gene expression. Because remember, that virus wants to hijack the machinery of your body, of your cell, and start making the proteins that the virus wants, not the proteins that you want. So it's saying that CBD is able to suppress or lower the SARS-CoV-2 changes in gene expression, which is good. If we can limit the gene expression changes, we can limit how much that virus can affect our body. Okay. So CBD was able to essentially eliminate the viral RNA expression in the host cells. This is so important. If we can eliminate the viral RNA expression in the host cells, it's not going to be able to propagate, reproduce, and actually spread more viruses throughout your body. So what gene expression are we talking about? It's the genes or the RNA code that codes for the spike protein, the membrane protein, the envelope protein, and the nucleocapsid proteins. So this is huge. It's essentially every major component of that of the SARS-CoV-2 virus. CBD was able to lower the RNA expression in the host cells. So your host cell saying, or the CBD is making your host cell say, nope, we do not want to produce those proteins of interest because something's off here, and we're going to just stop that right in its path so that it's not making proteins that we don't think are helpful for that cell. Excuse the personification. I tend to do that with everything. Okay. So let's just keep talking about these changes in gene expression. So both CBD alone and the SARS-CoV-2 virus alone are going to induce changes in the cellular gene expression. This is not surprising. We are introducing something from the outside world into cells that are only used to the inside of you. We're introducing that compound. It is probably going to change something in the gene expression. Sometimes that's good things, sometimes that's bad things. The question is, what is it in this case? So what's cool is when the cells were treated with the CBD, then infected with the SARS-CoV-2 virus, after that, they didn't return to the healthy baseline. So the CBD didn't help them return to this healthy baseline that they were at before they were treated with CBD. Instead, after those cells were pre-treated with CBD, then exposed to the virus, they returned to the levels that were observed when just CBD was given to those cells. So that kind of baseline of just that pure CBD, and this is like before even the virus was introduced. Some of the changes that were induced by the virus were then reversed by the CBD. One example of this was chromatin modifications. I'm not going to get in the details here because honestly, it probably doesn't mean much to almost everyone listening. But this is really big. The fact that CBD can actually reverse some of the changes that virus caused in our cells is huge. And we're going to talk about that more. Okay, a little recap because I think these are important. CBD will not prevent the virus from entering your cells. According to the last study, CBDA might, but CBD in this study won't prevent the virus from entering your cells. But usually when that virus enters your cells, it wants to hijack your cell's machinery and start to control which proteins are made because it wants to make the proteins that are coding for more viruses to be produced. The data here suggests that CBD can prevent the virus from making these viral proteins or expressing those viral proteins, and this will prevent the virus from propagating and spreading in your body. So that's pretty huge if it's true. And the current data says it's true. But remember, so far, we've only talked about cell data. Okay. So then the authors wanted to compare the data of the active compound that they're finding, CBD, compared to an inactive cannabinoid, which is going to be CBDV here. If you Google CBDV structure, you're going to see it looks very similar to CBD, but every little modification matters and it is slightly different. So CBDV was much less active in these assays compared to CBD. It did have some activity. I think it was around 60%, but nothing compared to CBD that was around 99%. So then the researchers compared the genes from the cells treated with CBDV versus CBD. So in theory here, if you say, okay, these compounds are so similar, but one of them is causing the effects that we want, CBD, which is preventing the viral replication of the genes, and the other one really isn't. So what is CBD doing that CBDV is not doing? The answer when they did their data analysis was essentially 3 sets of genes that were related to the endoplasmic reticulum were selectively upregulated by CBD, but not CBDV. I don't know if I know people talk about the mitochondria as the powerhouse of the cell all the time. I don't know if you guys remember the endoplasmic reticulum, but it has many functions in the body. Oh, in the cell specifically. So when the endoplasmic reticulum gets stressed out, it has too much to do, and the workload of protein folding is exceeded. That's one of its main purposes, protein folding. When that workload is exceeded, Sorry, my dad's calling and it just interrupted my video. Um, exceeded. Oops. Oh my gosh, I just got messed up. When this is exceeded, it it gets so overwhelmed that it sets off a signaling pathway. This signaling pathway is called the UPR, which is part of how your cells naturally respond to stress. Okay, I'm way too stressed right now. I'm going to set off the signaling pathway. That's what it's doing. This UPR is controlled by 3 proteins mainly, which are used to sense stress. When they are activated, when these proteins are activated, they will then go on to make changes to help control that stress and bring back the homeostasis, or just the balance that the cell normally has, that's functioning balance. CBD can induce these important proteins, specifically one protein called the IRE1 alpha, which controls the UPR. This is like extremely complicated when we get into the biochemistry of cells. It gets incredibly complex. But if you just remember that the IRE1 alpha is kind of this protein of interest that CBD can induce and manipulate, that is going to be the most important thing here. If you're crazy and you want to read more about this, I found this paper helpful. Again, if you just Google this, you can read it. But I wouldn't say it's exactly friendly for everyone to read if you're not into being biochemistry, science, heavy science, molecular mechanisms, whatever. Okay. So CBD can activate a stress response pathway in the cell, which aims to restore homeostasis or balance. That's just another recap. Okay. So the authors were saying, okay, our data suggests everything that I've been just saying, but it seems that there might be another way that CBD is working against the SARS-CoV-2 virus. And this is essentially, the data was saying, okay, we know that something's happening early in the infection process to make the cell unable to produce these proteins for the virus, but it's also effective later on. So what is happening later on in the infection process that's also making CBD an effective therapeutic, according to this paper? And this involves the innate immune system. So remember, you have like an adaptive immune system and your innate immune system. You're born with your innate immune system. It is used to protect you, to detect foreign matter, etc. Okay, so interferons are one way that the host immune system responds after pathogen exposure. So when something from the outside world, a pathogen, enters your body, the interferons are one way that your immune system tries to take care of that. It has been reported that the SARS-CoV-2 virus can suppress the interferon signaling pathway. We wouldn't want this pathway to be suppressed because this pathway is what is recognizing the foreign body and helping us combat it, essentially. So this is going to make our innate immune system worse at actually detecting that and getting rid of it. So they're finding that some interferon genes were moderately upregulated by CBD alone. So that's good. In theory, we would want to upregulate those interferon genes so that we can combat the virus. CBD alone can do that just a little bit. But what they found that was really cool is that the interferon genes were highly upregulated by CBD when in the presence of SARS-CoV-2, which is really amazing. And they found this in multiple places in this publication where, CBD alone could do something, but when it was CBD and the SARS-CoV-2 virus, it was a much more profound effect. I don't believe they know exactly the mechanism of why this is. So CBD could potentially lower the viral titer to enable the normal host activation of the interferon pathway. That's another just wrapping it up. Just make sure everyone knows the most important things. I don't know if you guys have heard, it's been actually in the news. It's been in multiple news stories about COVID. It's called the cytokine storm. So the cytokine storm happens later in the COVID-19 infection process, and it's responsible for a lot of the really, really bad effects that happen later on in the infection. So CBD can also effectively reverse the viral induction of cytokines. So cytokines are like small signaling molecules, but when they're powerful, like all initiated at once, this is what results in that deadly cytokine storm seen at the late stages of infection. If we can prevent this cytokine storm from happening, in theory, the turnout in the later stages of COVID after you've had it for a week or so, it's going to be a lot better. It's going to prevent people who are already, kind of worn out from fighting this virus. And then the cytokine storm comes along and kind of is like the last sweep. If we can prevent that from happening, in theory, the outcomes are going to be a lot better for COVID-19 patients. And just to reiterate, this was not seen with CBDV. It was only seen with CBD. What this paper does, what the last paper did not do, is they did use mouse studies. So mouse studies are known as in vivo studies, meaning in your body, in vivo, V-I-V-O. Compared to work that's done in a petri dish or a test tube, that's called in vitro studies. So in vitro studies have a lot of caveats. They're not usually translatable to animals or humans because we're so much more complex than what a petri dish can represent or a test tube. But these researchers actually did do mouse studies, which is a huge advantage of this publication. So they used mice that were able to express the ACE2 receptor protein, which again is what that spike protein on the COVID-19 virus is going to bind to cause infection. And they injected the mice with CBD 2 times a day for 7 days. This is kind of like pre-treating cells. They're pre-treating the mice. Then they infected the mice with the SARS-CoV-2 virus. And then they continued to dose the mice twice a day with CBD for 4 more days. They found that the CBD treatment significantly inhibited viral replication in the lungs and the nasal passages at day 5 post-infection in a dose-dependent manner. Why does it matter that we say in a dose-dependent manner? Because if it was not dose-dependent, it would mean that there was some other factor that was likely contributing to the therapeutic benefits that we're observing. When we say dose-dependent manner, this could mean like if you gave that mouse 1 milligram, it showed slightly improvement in the viral replication. If you gave it 2 milligrams, it showed a little bit better. If you gave it 5 milligrams, it showed about 5 times better than the 1 milligram, etc. Showing that as we increase that CBD, we also see that inhibition of the viral replication in the lungs and nasal passageways. So that is extremely good data for mouse studies. And again, if you want to see the details on that, check out the actual publication. Now, this can be a little misleading the way I've labeled it here, saying human studies, because these researchers weren't actually the ones conducting the human trials, because as I said, human trials can cost $1 billion, which no researcher in an academic lab is ready for that kind of study. But instead, I thought this was just so smart. The researchers knew that many people are already taking CBD daily for seizures, as I described before, with this pharmaceutical medication called Epidiolex, which is literally CBD. It's exactly the same thing. It is CBD. These people were taking it at 100 milligrams per day. So what they did is they essentially surveyed these people who have been taking CBD throughout the pandemic. And they looked back and were looking at the data of who got COVID, who didn't. So what they're finding is 75 people out of the 1,200 patients already using the CBD daily did contract COVID. That's about 6.2% of the patients in the study. They then tried to match this group of people with people who were not taking CBD daily. And again, this isn't totally controlled. They did the best they can with matching things like age, sex, etc., to get as best of a representation as they can, but it can't be exact. We're humans, and we're incredibly diverse. Even if on paper we look the same, in our body's biochemistry, our biochemistry is different. Okay, anyway. But what they did find is when they tried to match this with people who were not taking CBD every day, with the match control group, they found an 8.9% positivity rate. So this is significantly higher in the other group. This data, should be taken with a grain of salt just because it's not exact, but it's really It's cool data. It's cool that they wanted to show that this was translatable to humans and to animals and to cell assays. And I thought it was a really clever part to put into the publication. We got a few slides here on the conclusion. So CBD induces this IRE1 alpha gene. This can potentially account for the ability of CBD to degrade the viral RNA and to induce interferons by the RNA fragments, thus activating the human innate immune system. So that's a pretty complex sentence, but essentially saying the activation of that gene alone by CBD could be responsible for both ways that CBD works against the COVID-19 virus. And it's working against your innate immune system by activating those interferons that could be suppressed when you're just infected with COVID and not taking CBD, and also for essentially preventing that viral RNA from producing the proteins needed to replicate. CBD can suppress the cytokine activation in response to viral infection, reducing the likelihood of immune cell recruitment and the cytokine storm within the lungs and infected tissues. Again, this is an endpoint in the COVID-19 infection that people have been trying to limit for a long time, because this is when patients really start to show really bad signs of severe COVID-19 infection. If we can prevent that cytokine storm, even if CBD was ineffective in everything else this paper is claiming, but it was effective against preventing that cytokine storm, this is still huge data. CBD has the potential to help in early stage and in late stage infections by protecting against the overactive immune system. Similar to the way that cannabinoids work in our body at bringing back homeostasis through the endocannabinoid system, it seems like that's kind of what CBD is doing here, but through different mechanisms. Essentially, if things are too active or if proteins are being produced that shouldn't be produced, the CBD's inducing genes that are able to shut that off. Or in the case of the cytokine storm, if we want to prevent that absolute swarm of recruitment of the cytokines and these inflammatory markers, CBD is able to prevent that cytokine storm from happening. Again, this is really important. CBD is generally regarded as safe. Even with other natural products that we've been testing against the SARS-CoV-2 virus, not everything is safe. Not everything from nature is safe, and not everything that's synthetic is not safe. And because we know that CBD is generally regarded as safe and tolerated in very high doses, we know that this could be a real solution towards preventing very bad COVID-19 infections. CBD only has minor side effects, and one of those side effects is the interaction with other drugs in your body, which I say on the next slide. But if you're taking other medications, something like antidepressants, anti-anxiety medication, and hundreds of other medications, it should be noted that CBD can interact with those drugs and could either prevent the efficacy of those drugs or prevent the metabolism, which could cause the buildup of certain drugs in your system, which you don't want. So it's always recommended that you talk to your doctor before starting everything, even something like CBD, especially in high doses. CBD can be taken orally, unlike other current COVID-19 treatments, which is huge. We don't want to have to IV inject treatments into every single patient. If you could do this at home, that would be very helpful for hospital staff who's been risking their lives since day one. Very important is that the metabolite of CBD is also active. Again, if you're taking a tincture, an edible, whatever of CBD, if you have COVID, you probably don't want to smoke a joint, blunt, bowl, bong, whatever, because your lungs are already compromised at that stage. So you're probably taking this orally. And if you're taking it orally, it's going to be metabolized in your liver. And one of those metabolites is 11-hydroxy-CBD. And it's really great to know that that metabolite is also active, which is just adding to the potential of using this as a treatment. CBD can be used prophylactically, meaning as a preventative treatment in a way. I mean, again, we need more data to say that for sure, but this is very important when thinking about stopping the progression of this virus from spreading even more rampant across the world. CBD works after the virus enters the cell. In theory, this would make it effective for new variants as well, because the spike protein is typically what mutates. So in the last paper, they were talking about how they were targeting the spike protein and the ACE2 receptor for preventing the COVID-19 virus from entering cells. The issue there is the spike protein is usually what we see mutate, which is going to be an issue for the acidic cannabinoids highlighted in the last paper. But here, the CBD is not preventing these compounds from entering the cell. Instead, they are entering the cell, but the CBD is preventing then I've said this a million times, the genes from being transcribed and the proteins from being produced for actually allowing that virus to infect our bodies. So this could be huge news for when a new variant comes along. It's likely that CBD would still be effective, according to the data presented in this paper, at preventing further infection. The authors also suggest cannabinoids like THC could interfere with the efficacy of CBD. So THC was not found to be active, and many other cannabinoids that they were testing were not found to be active. So they're saying that THC could potentially interfere with how well CBD works in the body. They don't really have data on that, but I understand where they're coming from, because this is kind of the classic approach to drug discovery, is if you know one thing works really well, don't add in other variables like other cannabinoids that could potentially cause adverse effects or cause some other molecular mechanism to be set in place, which would offset what the CBD is doing. Also, just take note, again, a high dose of CBD is needed in this study, but these authors did demonstrate that it is possible to get the plasma levels to that concentration, which is huge. They also emphasize that high quality and high purity CBD is needed. They suggest against using flavored CBD products like peppermint, mango, things like that, because that will decrease the purity. Some CBD might say on the vial like 99% pure, something like that. That's what you're going to want to look for if you wanted to do anything related to this study. And as I just mentioned, if you're taking other drugs, CBD may interfere how these drugs work or are metabolized in the body. Okay, last slide. What are the key differences? I've kind of been saying these as I went. What are the key differences from the last publication that I talked about in the last video and this publication? And the last publication was titled Cannabinoids Block Cellular Entry of SARS-CoV-2 and the Emergent Variants. I mentioned there are various druggable targets in the infection of the SARS-CoV-2 virus. These publications do not compete with each other's accuracy, and they don't invalidate each other by any means. They're looking at completely different things. They're testing different endpoints. So in no way am I invalidating the last publication with what I'm saying with this one. The last publication targets compounds from cannabis that interact with a portion of the spike protein, which again, this publication is not looking at. We know that in this case, CBD isn't working until that virus has entered the cell. Something I really liked about the last publication, which this publication did not do, is the last publication seemed a little more unbiased, because they were looking at a panel of cannabinoids and they said, oh, wow, these acidic cannabinoids work the best. This publication seemed to really emphasize that CBD was the star of the show here and that they tested other ones just to make sure that they weren't as active, but it wasn't really a screening paper. I love screening papers. I'm biased because that was my dissertation. But anyway, the last publication found that the acidic versions of the cannabinoids were the most active. This version is not saying the acidic versions, it's saying the non-acidic versions. So if you were to be smoking a product with CBD in it, that would be the active compound because it has been decarboxylated. Again, please see the last video for a review on decarboxylation. And on that note about smoking, again, this author is saying that you should not be using essentially complex formulations like those found in flower that have hundreds of different compounds, dozens of different cannabinoids, terpenes, flavonoids, etc. This publication saying Use high quality pure CBD isolate, just the CBD. Don't include other cannabinoids. The last paper was saying, yeah, you might have better results if you use a diversity of different cannabinoids, but the acidic versions of CBDA and CBGA are going to be the most effective. The last publication by Bremen and colleagues did not show any animal studies, so no in vivo work, and they didn't draw any conclusions about human studies either, which was very smart of them for the data that they had. So the current study I'm describing is a little bit more translatable to humans because they tested it in animals and they had the data from humans. The effective doses were much lower in this study compared to the Bremen study, which shows that it is achievable because we had data from people taking Epidiolex, which is CBD. Other than that, that's pretty much the main differences between these two publications. Altogether, I take CBD every day as it is, but I think I'm going to start taking it even more because honestly, I find this data very compelling. But it is important to note these data are still not clinical trials, human clinical trials, but I do believe that the data presented in this publication will provide enough background and enough data to now facilitate human trials with CBD as a preventative against COVID-19. Wow. Please subscribe to this channel if you find this interesting at all. And then please comment below if you want me to cover different publications or if you have questions on this one. Thanks always for listening.